Our next speaker is Dr. Barbara Cook. Barb is a senior lecturer at UWA and a biodiversity scientist with extensive research and teaching experience in the taxonomy, ecology and genetics of aquatic biodiversity and the ecology and management of rivers and wetlands. She has a particular interest in Mediterranean systems and relictual Gondwanan fauna. Barb is presently leading a project on the projections of impacts of climate change on biodiversity in southwestern Australia and South Australia, including species distribution modelling, climate vulnerability analysis and the use of spatial data for planning. She's also a great supporter of South Coast NRM, and we really appreciate her involvement in getting this conference up. Thanks, Barb.
Thank you, Penny. So, and thank you very much to all those other speakers that have gone before me, because you've prepared the ground somewhat, which has been very useful for me. So this talk, we'll talk a little bit about climate change, but I'm going to throw in a couple of other threats as well. And although we won't be looking specifically at the interactions between these threats, I'll just show you a project that we ran with SWIC, the Southwest Catchment Council, where we developed and applied a framework for assessing the vulnerability of aquatic species to multiple threats. I must confess I'm particularly fond of aquatic species. So the idea was that we wanted something that was simple, a points-based framework which we could then use to assess. To start off with, we looked at aquatic species, but of course you could actually do this for terrestrial species as well. And we wanted to look at more than just climate change, because obviously there are other threats which are really important. And then we wanted to, having developed this framework, we decided we would actually apply it to a particular catchment. And yes, I know that Blackwood is not in the South Coast, but of course we could apply this to any of our systems as well in the South Coast region. Very basically, this is what it looks like. We looked at 3 threats where we were able to get data for multiple species across the whole of southwestern Australia. And those were climate change, secondary salinisation, and nutrient enrichment. So these are 3 key threats that operate in the aquatic environment. Obviously, there are a lot of other threats as well, but these are probably the ones, the universal ones that you'll see in all our wetlands and rivers. And then what we did was we used species distribution modelling, but that is actually really only an indicator of exposure when we look at climate change. So we looked at it, when we look at vulnerability of species, we looked at exposure, sensitivity and adaptive capacity. And a number of speakers have picked up on bits and pieces and have mentioned that, of course, we have no idea how species are actually going to be able to adapt and whether they're going to be particularly sensitive. Now, I'm not going to go through all these 9 indicators that we've scored for hundreds and hundreds of species for southwestern Australia, but I will focus, because this is a climate change conference, I'll focus on the 3 indicators that we used for climate change. Perhaps before I move off, there are a couple of other abbreviations that I need to tell you what they are. So for each of the 3 threats, if you look down the right-hand side, you'll see CCVI, SVI, NEVI. Those are vulnerability indices for those specific threats. And then they're added together to get a standardized overall threats vulnerability index, which is standardized to a value of 100 in this particular example. So, as promised, I'll have a closer look at the climate change one. So for exposure, we looked at the change in bioclimatic envelope. So this is where the species distribution modelling came into our project. For sensitivity, we looked at thermal tolerance. So that is, in the case of the invertebrates, we gathered as much data as we could on the upper thermal tolerance of the invertebrates. And in this case, one can get data on laboratory experiments as well as ecological field data. And so we used that for the invertebrates. In the case of the fish, the freshwater fish, there are very few data available for laboratory type data for upper thermal tolerance. So we actually had to use ecological field data for the fish. So that's thermal tolerance then for sensitivity. And then for adaptive capacity, we looked at the dispersal ability of these invertebrates and freshwater fish to see whether they'd actually be able to move. Okay, so for exposure then, species distribution modelling, and I'm not going to go into details about the models and the RCPs, etc., because of lack of time, but very basically, we looked at the percentage change in the bioclimatic envelope, and then we scored each species, either a 1, 2, or 3, according to the reduction in the bioclimatic envelope. So a score of 1 then, if the percentage change was less than 25%, between 25 and 75% reduction, a score of 2, and if there was more than a 75% reduction in the bioclimatic envelope, a score of 3. It's a very simple process that we followed. And here's an example. You've seen a couple of, Sean has kindly shown you some of the maps that we get from bioclimatic modelling. If you look at the left-hand side, your left-hand side, that's the modelled distribution. So that's the bioclimatic envelope where red is most suitable for this particular fish, which is the black-striped minnow, Galaxiella nigrostriata. And then on the right-hand side, the modelled distribution going into the future, 2080, business as usual carbon emissions, so that's the RCP 8.5, and what this amounts to is an 88% reduction in bioclimatic envelope. So that particular species gets a score for that indicator of a value of 3. Sensitivity for thermal tolerance for the 11 freshwater fish species that occur in southwestern Australia. And these are, I know there are more than 11 fish that you can find up river systems, but these are the freshwater fish. So these are fish that are of freshwater origin, so they're not of estuarine origin. Same story, we looked at the temperature at the sites of occupancy, the mean temperature, and had a look at the data for that. So there's many data points, and scored them according to the mean temperature at their sites of occupancy. So the example that I'm just tracking here, which is that black striped minnow, you'll see that it has a value of 20, and that gave it a sensitivity score of 1, because you'll see for below 17 degrees, highly sensitive, 17 to 19, a score of 2, and more than 19 gets a score of 3. Adaptive capacity, similar sort of way of going about it. Small, in this case, a small species, very localized in terms of migration ability, and so that particular species that I'm tracking, the yellow one there, the black-striped minnow, received a score of 3. Now, we did this for hundreds of species. Okay, so we're able to get some scores of their threats, their vulnerability, and work out vulnerability indices. Now, I want to turn to the system that we applied this, which is the Blackwood River system. So to do this, you need to gather as much information as you can about the system that you're going to apply the framework to. So the little green dots there indicate the sites where we were able to get data on the species that live there, as well as some environmental data for that particular case study. And now for the Blackwood, we managed to actually score 100 aquatic invertebrate species and 8 freshwater fish species. I do need to digress very briefly here. Now, although I'm not going to look at how we scored the nutrient enrichment and the salinization in the framework, what I do need to point out is that to actually score exposure to salinity and nutrient enrichment, that is done at a localized level. So what we do is we look at the proportion of records of the selected species within the given catchment. So obviously in this example it's the Blackwood River catchment. And then we look at the proportion of those records that occur at a value that is above a threshold value. So I'll show you very quickly. The threshold values actually come from the literature and from various other studies that tell us at which values these animals are going to have problems. So in the case of salinity, it's around about 3 parts per 1,000. Total nitrogen, a value of 1.2, and total phosphorus, a value of 0.08 milligrams. And then what we do, same story, 1, 2, and 3, depending on what proportion of your records occurs in water that exceeds that value. So very simply, this is just the example of the salinity. Using all the data that we can get hold of for that particular system, we work out for the whole catchment which parts of the catchment occur above the threshold value, and then we look at the proportion of records at that threshold value. Okay, so let me very quickly summarize what we found for the Blackwood. 3 things I'll just quickly focus on. Firstly, applying this framework, we identified the most vulnerable species that actually occur in there and which would be worthy of continued conservation efforts and possibly recognized as flagship species. And I'll show you the graph. What we found was about 70% of the species that we assessed, so 70% of the 108 species, were considered to be of relative low vulnerability in terms of their total vulnerability index. About 27% were of moderate vulnerability, and applying this framework, only 1 species actually came out to be of high vulnerability, which is quite interesting. And that's this little salamander fish. So that's, of all the 108 species, that's the only one that really came out as being possibly in a spot of bother. The second finding I just want to point out is that we were able to look at the relative importance of the 3 threats that we were looking at. They're fairly similar, although statistically, secondary salinization, the vulnerability index for secondary salinization was significantly higher than that for climate change and nutrient enrichment. Tells us that perhaps we shouldn't take our eye off the ball when it comes to secondary salinization. And then the third thing we were able to do was to map the spatial distribution of the concentrations of vulnerable species. And what we find is that in this case, in our case study, the concentrations of vulnerable species is actually in the lower part of the catchment for various reasons. And the red areas are where you've got concentrations actually of salamander fish in this case, and then the orange would be a number of those other species that actually are vulnerable. So we've successfully developed this framework. We applied it to the Blackwood River. Those are the 3 things I've just gone through. And most importantly, this framework can now be used. If we wanted to, we could actually use this for any system in the southwestern Australia. So we could, for example, do a number of the systems in the South Coast region. Thank you.